In order to view a specimen under a microscope, it has to be first placed on a clean glass slide. Placing the specimen on the slide is called mounting. Two types of mounts, dry mount and wet mount, are generally prepared in the laboratory based on the type of specimen to be viewed. A dry mount is generally used for viewing inanimate or non-living objects like pollen, hair, feathers, etc. It does not require water for mounting. A wet mount is generally used for viewing animate or living objects such as organisms and cells. Wet mounts are prepared using water or glycerol. In a wet mount, a small piece of the specimen is placed at the center of the slide with 1 or 2 drops of water or glycerol. The specimen is then covered with a cover slip and observed under a microscope. In case of permanent slides, that is, the slides that need to be preserved for future use, materials other than water, for example Canada balsam, need to be used. The specimens to be observed under a microscope are often stained with a dye. Safranin, methylene blue, and crystal violet are some of the commonly used staining dyes. Staining procedure highlights the biological tissues and specific regions in the cells. This makes it easier for us to view the details of the specimen. Experiment 4. Microscopic organisms in the water. The aim of the experiment is to observe the microorganisms present in the drop of water under the microscope. Come, before we begin with the experiment, let us take a quick look at the materials required to perform this experiment. The materials required to perform this experiment are beakers, glass slides, cover slips, dropper, mounting needle, blotting paper, and a compound microscope. Let us now begin with the experiment. First, we will collect water samples from a sea, river, and puddle in a beaker and will note down the place of collection of the sample on the label pasted on the beakers. Now, with the help of a dropper, we will take few drops of water from the beaker labeled as sample 1C and transfer it onto the glass slide. Cover it with a cover slip using a mounting needle and clean the slide with the help of a blotting paper. Come, let us observe this slide under the low power objective of a compound microscope. What do we observe under the microscope? We see many microbes moving about in it. Algae and bacteria are seen under the microscope. Now, we will prepare a slide for sample 2, river, in the similar manner as we did for sample 1. Come, let us observe this prepared slide under the low power objective of a compound microscope. Students, what do we observe under the microscope? We can see many amoeba and paramecium moving under the microscope. Come, let us now prepare a slide for sample 3, puddle. and view it under the low power objective of a compound microscope. When we observe this slide under the microscope, we see bacteria, amoeba, and algae in it. Come, let us draw a diagram of bacteria, amoeba, paramecium, and algae that are seen under the microscope. Thus, after examining all the samples, we can infer that different types of microorganisms like bacteria, amoeba, paramecium, and algae are seen in the water collected from different locations. Remember, there may be many microorganisms or microbes in a drop of water, though they are not visible to our naked eye.